every single second that you are listening to this, your body is churning out like roughly 2 million red blood cells.
It is a staggering number, yeah.
Right. It is this massive lifelong assembly line that literally never takes a break. But the really bizarre part, the master cells responsible for all this production, like the absolute origin point of your entire blood system, they actually spend almost their entire lives completely asleep.
Which is so counterintuitive when you think about how much blood we need.
Exactly. And understanding why they sleep and why they eventually, refuse to wake up as we get older, well, that might actually hold the key to reversing the way our immune systems age.
We are definitely looking at a fundamental shift in how we understand human regeneration here, because for decades, the biological machinery behind how our blood is made has been, frankly, incredibly elusive.
Like, we knew the factory existed, but the blueprints were just sort of smudged.
Right.
Which perfectly brings us to the mission of our deep dive today. We are exploring this cutting-edge study that was just published in Nature Communications in March 2025. It's titled Continuous Map of Early Hematopoietic Stem Cell Differentiation Across Human Lifetime.
It is a very dense, very exciting paper.
It really is. And our goal today is to map the exact birthplace of human blood, uncover what happened to our most vital stem cells as we get decades older, and reveal this newly discovered flag that researchers can now use to pinpoint the purest, most protected stem cells in your body.
And I think the sheer scope of this research is what makes it such a massive leap forward. Previous attempts to map this process were essentially looking at like a blurry satellite image of a continent.
Just big shapes, no details.
Exactly. But this new research provides us with street-level maps for the very origin of our blood system. They tracked individual cells across the lifespan of healthy donors.
Okay, so let's unpack this, starting with the factory itself. So the technical term for this whole process is hematopoiesis.
Right, hematopoiesis.
And you can think of it like this sprawling, highly secure manufacturing plant hidden deep inside your bone marrow. In this plant, you have one ultimate raw material, a true stem cell.
The holy grail of the blood system.
Exactly. And that single, totally unassuming raw material has the magical potential to transform into hundreds of highly specialized tools. So it can become the red blood cells carrying oxygen to your brain, or the white blood cells hunting down a virus, or, the platelets that rush to clot a paper cut.
Right, and the scientific name for that ultimate raw material is a hematopoietic stem cell, or HSC. But the persistent hurdle the scientific community has faced is actually finding the purest form of that raw material.
Because this is a huge mess in there.
Oh, it's a completely chaotic soup. Isolating a true uncommitted stem cell from the bone marrow has historically been a massive challenge.
The paper actually calls this the impurity challenge. So to give you an idea, even when researchers use our best current methods, which basically involve looking for specific protein markers on the outside of a cell, like, say, finding a cell that waves a CD34 flag but drops a CD38 flag, the batch of cells they pull out is still wildly inconsistent.
Yeah, and that's a huge problem because when you isolate cells using those standard surface markers, you just assume you're gathering a pristine batch of raw stem cells.
Right.
But if you put those extracted cells to the test in specialized models, like asking them to actually regenerate a complete long-term blood system, the reality is incredibly stark. Only about 10% of them are actual true stem cells.
Wait, only 10%. So what are the other 90%?
They're basically imposters. I mean, they might look like stem cells on the outside waving the right flags, but internally, they have already started to specialize. They've quietly lost that multi-purpose potential.
Okay, let's pause and do a quick recap of that because that is wild. So researchers have been trying to study the origin of blood for years, but 90% of their supposed stem cell samples were already tainted. They were already secretly deciding what they wanted to be when they grew up.
Exactly. The blueprints were ruined from the start.
So how did the team behind this new paper finally cut through the noise and, solve that purity problem?
Well, they realized they had to move beyond just looking at the outside of the cell. So they used this technological approach called single-cell proteo-transcriptomic sequencing.
That is quite the mouthful.
It is, yeah. Specifically, they used a platform known as BD Rhapsody. What this means in practice is that they didn't just blend up a bulk sample of bone marrow and look at the average. They isolated over 62,000 individual bone marrow cells.
62,000. One by one.
One by one, from 15 healthy human donors spanning various ages.
I mean, analyzing 62,000 cells individually is just an immense amount of data.
It is mind-boggling. And for every single one of those cells, they measured 2 entirely different layers of information simultaneously.
Okay.
First, they looked at the surface proteins, so 46 different physical flags on the cell's outer membrane. But second, they looked deep inside the cell at the mRNA to measure the expression of 596 targeted genes.
Oh, wow. So they were basically reading the internal genetic diary of every single cell while concurrently scanning its external uniform.
Exactly. They cross-referenced the outside and the inside simultaneously, and by overlapping those 2 massive data sets, they finally found the absolute anchor point of human blood.
The purest of the pure.
Right. They isolated a very specific subcluster of cells, and they named it HSC1.
HSC1. Okay, so what actually defines this elite little group? What is in their genetic diary?
The defining characteristic of the cells in this HSC1 cluster is a state called quiescence. They are profoundly dormant.
Like deep sleep.
It's very deep sleep. When you look at their mRNA, the genes responsible for the cell cycle, the engines that drive a cell to divide, grow, and multiply, they're completely powered down.
But they aren't dead, obviously.
No. While they're resting, they are actively hoarding a very specific set of genes designed to maintain their raw potential.
So what exactly is keeping them in this deeply dormant state? Like, is there a specific genetic switch they're pulling to stay asleep?
There are a few, but a really critical one they identified in the data is a gene called HLF, which stands for hepatic leukemia factor.
HLF.
HLF is crucial for defining this true stem cell state and literally locking the cell into that protective dormancy. By pinpointing this HSC1 cluster and the role of genes like HLF, we finally have a true molecular blueprint of an untainted blood stem cell.
Before it makes a single decision about its future.
Exactly.
Okay, so that is our first big takeaway. We found the true anchor, HSC1, and it's kept asleep by genes like HLF. But now that we have the starting point, the obvious question is, what happens to these dormant cells over time? Because they didn't just look at young donors, right?
Right. They looked at people across a whole lifespan. And to make sense of how a cell ages, the paper uses a fascinating concept called a pseudotime trajectory.
Pseudotime trajectory. Break that down for us.
So a pseudotime trajectory is a continuous mathematical map. Instead of just taking a static snapshot of a cell, you use the sequencing data to algorithmically order the cells along a timeline of development.
Like turning photographs into a movie.
Yes. You can literally watch the progression from a raw HSC1 stem cell to a mature, functioning blood cell.
It's sort of like tracking a student from their first day of kindergarten all the way through college and mapping exactly when they decided to choose a specific major.
That captures the concept perfectly. And the trajectory reveals that the first major branching point for these stem cells, like the very first definitive fork in the road, is the decision to move into the megakaryocyte erythroid lineage.
That's a big word, megakaryocyte erythroid. That's the pathway dedicated to churning out red blood cells and platelets, right?
Exactly. That's the baseline survival stuff.
But looking at the trajectory data across different ages, I noticed a glaring paradox in the study.
The aging paradox.
Yeah, because the study compared young adults around age 22, middle-aged donors around 60, and older adults around 75. And the data clearly shows that the older donors had a significantly higher raw number of these immature HSC1 stem cells just sitting in their bone marrow.
Which throws everyone off.
Right. If older people have more of the ultimate raw material, shouldn't their blood and immune systems be way more robust than a 20-year-old's?
You've hit on the fundamental aging paradox in hematopoiesis. Our intuition suggests that having a larger reserve of stem cells should equate to immense regenerative power.
Yeah, more raw material equals more products.
But the data reveals a totally different reality. The structural pathways of the factory remain consistent over a lifetime, but the biological traffic flow undergoes a radical shift.
A traffic flow shift. So the older stem cells are just piling up at the start of the assembly line. Why are they getting stuck?
The mechanism comes down to a failure to transition their gene expression. We mentioned earlier that young, healthy cells are efficient, right? They wake up, move down the line, and mature.
Right.
But the cells in older donors get trapped hoarding the genes that keep them dormant. And the paper identifies a major culprit here, a gene called DLK1.
DLK1. So how does DLK1 actually force a traffic jam? What is it doing physically inside the cell?
DLK1 acts like a biological brake pad on the cell's metabolism.
A brake pad. Okay.
Yeah, because for a stem cell to mature and differentiate, it has to undergo a massive metabolic shift. It basically has to ramp up energy production to fuel its transformation into a specialized cell.
It needs to turn the engine on to drive down the highway.
Exactly. And DLK1 restricts that metabolic engine. It forces the cell to remain in a low-energy, dormant, stem-like state.
Oh, wow. So as we age, our stem cells are overexpressing DLK1. They are essentially clinging to their youth by refusing to turn on their engines, which prevents them from ever growing up.
And the downstream consequences of that are severe. Because the cells at the start of the line refuse to mature, there is a drastic drop in the production of functional adult cells at the end of the assembly line.
They're starving the rest of the factory.
Right, and the paper highlights that this specifically starves the lymphoid lineage. That's the pathway responsible for creating T cells and B cells.
Which perfectly explains why older individuals often struggle with weakened immune systems.
Exactly.
Their bone marrow is literally packed with viable stem cells. The raw materials are all there, just sitting quietly. But because of this DLK1 brake pad, they stubbornly refuse to transition into the immune cells the body desperately needs to fight off new infections.
The systemic failure isn't a lack of supply. It is a failure of the developmental pipeline. The cells are locked in a state of suspended animation.
Let's do another quick check-in here, because this is crucial. If you're listening, imagine you are one of these aging stem cells. You've been sitting in the bone marrow for like 6 or 7 decades. You're completely asleep. Your engine is turned off by DLK1, and you're just physically piling up with millions of other stuck cells.
It's a very crowded waiting room.
Right. And it feels like this deeply dormant state would make these cells highly vulnerable to the wear and tear of time, specifically genetic mutations.
And that transition to genetic mutations brings us to a really crucial twist in the study involving a condition known as clonal hematopoiesis, or CH.
Clonal hematopoiesis. That is one of those conditions that becomes a major risk factor as we age.
It does.
As I understand it, it happens when a single mutated stem cell gains an unfair survival advantage, starts cloning itself out of control, and just slowly takes over the blood supply. It's frequently a precursor to severe blood cancers like leukemia, right?
That's exactly it. Over the course of a human lifespan, our DNA inherently accumulates errors. Background radiation, metabolic byproducts, replication mistakes, they all contribute.
It's unavoidable.
Yeah. And certain driver mutations can give a rogue cell the ability to outcompete healthy cells.
So this is where I get incredibly curious about that pure HSC1 cluster. If these HSC1 cells are the deepest, purest reservoir of our blood, and they are sitting around for decades in older people, are these dormant cells the origin point for all these dangerous mutations? Like, are we actually hoarding mutated monsters?
The findings here are completely counterintuitive.
Oh.
The research team zeroed in on the most notorious driver gene for clonal hematopoiesis, a gene called DNMT3A.
DNMT3A.
Okay.
It is by far the most frequently mutated gene associated with this condition.
Yeah.
But when they analyzed the ultra-pure HSC1 cells, they found that the expression of DNMT3A was virtually undetectable.
Wait, what? The gene responsible for the most common aging blood mutation is completely silent in the oldest, purest stem cells. It is entirely turned off in the HSC1 cluster. The data reveals that these leukemia-associated genes only activate, and therefore only become problematic, further down the assembly line.
So they're safe while they sleep.
Yes. The trouble doesn't start until the cell leaves the HSC1 stage, wakes up, and reaches what is called the MPP stage.
MPP, that stands for multipotent progenitor, right?
Correct.
So this is the stage where the cell has finally taken a step out of bed, shaken off the DLK1 brake pad, and is actively trying to grow up.
Exactly. And to understand why the mutation only matters then, we have to look at the mechanism. Most DNMT3A mutations are what we call loss-of-function mutations.
Meaning the gene breaks and stops doing its job.
Right. But the cell actually needs the DNMT3A gene to function properly in order to successfully navigate that MPP stage and differentiate.
Okay, let me try an analogy here. It's like having a car with a completely broken steering wheel.
Okay.
If the car is just parked in the garage, which is our dormant HSC1 state, a broken steering wheel doesn't really matter. You wouldn't even know it was broken.
Right, you're not going anywhere anyway.
Exactly. It only causes a catastrophic crash when you finally turn on the engine and try to drive it down the highway at the MPP stage.
That analogy captures the mechanism perfectly. The loss of function only triggers a crisis when the function is actually required.
That is fascinating. But what does this revelation actually mean for how we treat these diseases?
It is huge. It provides the medical community with an incredibly precise temporal roadmap. For a long time, the assumption was that to cure these conditions, we had to target the absolute top of the hierarchy, the most immature stem cells.
Burn it out at the root.
Right, but this single-cell data proves exactly when the genetic machinery breaks down. If a pharmaceutical company is developing a targeted therapy for leukemia, they now know the DNMT3A mutation isn't driving the pathology at the HSC1 stage.
The target is at the MPP stage.
Exactly. That shifts the entire paradigm for precision drug development.
So we have this beautifully detailed map now. We know the anchor point is HSC1, we understand how the DLK1 traffic jam starves the aging immune system, and we know exactly when mutation traps like DNMT3A trigger a crisis.
That's a very complete picture.
But there is a massive practical problem here, isn't there? Because reading a cell's internal mRNA, which is how we found HSC1, that requires breaking the cell open and destroying it.
It does.
So how does a doctor or a researcher actually catch one of these ultra-pure HSC1 cells in real life, keeping it alive to use in medicine?
And that is the crucial translational step, because transcriptomics is phenomenal for analyzing a cell after the fact. But to leverage these cells in regenerative medicine or bone marrow transplants, you need a way to extract them from a living patient intact.
So how do they do it?
This is where the team's dual sequencing approach pays off. Remember the 46 surface proteins they mapped?
The physical flags on the outside of the cell, yes.
Right. They scoured that massive database. They were searching for a unique barcode, a single flag that is planted almost exclusively on the pristine HSC1 cells, but vanishes the moment the cell starts to wake up.
And did they find it?
They did. They found a surface protein called CD273, which is also known in immunology as PDL2.
CD273. So this is the ultimate purity flag.
It is. And isolating CD273 as the definitive marker is a monumental finding. But to validate it, they took living bone marrow samples and used fluorescent antibodies to sort the cells into two groups.
Okay.
Those with high levels of the CD273 flag and those with low levels.
And from the study, they took those two groups, placed them in a dish, and put them under continuous video microscopy to watch their behavior in real time. I mean, the visual evidence must have been striking.
Oh, the video tracking completely confirmed the sequencing data. The cells waving the CD273 flag took significantly longer to undergo their first cell division compared to the CD273 low cells.
They were actively hitting the snooze button.
Absolutely. Furthermore, when they analyzed the isolated CD273 high cells, they found drastically reduced levels of a gene called CDK6.
CDK6. What does that one do?
CDK6 is a primary enzymatic driver that forces a cell into the active division cycle. The absence of CDK6 provides mechanical proof that these CD273 high cells are deeply entrenched in their dormant state. They truly are the truest stem cells we have ever isolated.
Okay, but discovering CD273 isn't just about finding a convenient handle for scientists to grab onto, is it? There has to be a deep evolutionary reason why these specific stem cells are waving this specific flag.
And you've hit on the most beautiful part of the biology here. The function of CD273 goes far beyond being a structural marker. CD273, or PDL2, is a highly potent immune-modulatory molecule.
Wait, so it's interacting with the immune system. It's acting like a VIP bouncer for the cell, or like a giant biological do not disturb sign hung on the door.
It is an active defense mechanism. CD273 interacts directly with the body's immune system. Its primary function is to suppress T-cell activation and block the release of inflammatory cytokines in its immediate vicinity.
Okay, let's play out why a stem cell would need to actively suppress the immune system. Because that sounds weird at first.
It does.
Imagine a scenario where your body contracts a massive systemic infection. Your immune system just goes nuclear. T-cells are swarming, inflammation is spiking everywhere, your internal environment basically becomes a toxic war zone to fight off the pathogen.
A very dangerous environment for any cell.
Right. And if those highly aggressive T-cells accidentally targeted and destroyed your deepest reservoir of dormant stem cells during all that chaos, you would permanently lose the ability to generate new blood.
The consequences would be fatal. You wouldn't survive the recovery.
Wow.
So our deepest, most valuable stem cells have evolved this CD273 shield specifically to prevent themselves from becoming collateral damage during an immune crisis. By waving this flag, they force the immune system's riot police to stand down and walk away.
That is incredible.
They create a localized zone of profound immunosuppression so they can sleep safely through this systemic storm.
They are surviving by hiding in plain sight. Yeah. Literally actively telling the body's own defense mechanisms to just look the other way.
It is a breathtaking example of evolutionary elegance.
Okay, so let's zoom out and recap this incredible journey for everyone listening. We started with a messy, undefined soup of bone marrow. We identified the absolute anchor of human blood in the HSC1 cluster.
Driven by HLF.
Right. Then we saw how aging creates a metabolic traffic jam, thanks to that DLK1 break pad-trapping these cells and starving our aging immune systems.
The aging paradox.
Yeah. And then we discovered that dangerous mutations, like DNMT3A, they just lie in wait until a cell tries to mature at the MPP stage. And finally, we uncovered the CD273 shield that our body uses to guard its most precious regenerative assets.
Exactly.
So why is mapping this specific pathway so urgent for the future of medicine? Why should the listener care?
Because the clinical applications are immediate. By utilizing the CD273 flag to isolate purely multipotent immune-protected stem cells, we are looking at a total paradigm shift in bone marrow transplantation.
How so?
Well, imagine the difference between giving a leukemia patient a transplant where 90% of the cells are already compromised imposters trying to specialize versus providing a transplant of 100% pure, true stem cells that already possess a built-in shield against immune rejection.
It elevates regenerative medicine to a level of precision we just haven't had before.
We are no longer throwing a handful of random seeds into the soil and just hoping one takes root. We can now plant the perfect seed in the perfect environment.
That precision literally changes outcomes. It saves lives.
It absolutely does.
Which leaves us with a final, honestly somewhat unsettling thought to ponder today. We now know that CD273 is the ultimate do not disturb sign that protects our pure stem cells from immune destruction. But in biology, powerful shields are rarely a secret for long. So it begs a darker question. Is this the exact same invisibility shield that mutated blood cancer cells eventually hijack to hide from our immune system?
It's a very real possibility.
Right. Because if we figure out how to temporarily lower that CD273 shield to wake up our aging stem cells and cure the immune traffic jam, do we accidentally open the door for hidden leukemias to strike? The line between protecting our youth and shielding a disease might just be a single protein pick. Until next time, keep diving deep.